Welcome to the deep dive. Our mission here is, well, pretty straightforward. We take your stack of sources, articles, research papers, maybe notes you've gathered, and we unpack the most important bits of knowledge, the key insights hidden inside.
We're here to be your guides, really, help you cut through all that dense information and zero in on what actually matters.
Mm-hmm.
And today, we are plunging into a really fascinating stack of material. It's largely centered around the DevoWorm project.
Right.
And these sources tackle a huge question. How are scientists using computational approaches?
Mm-hmm.
I mean, everything from physics-based models to really cutting-edge machine
To
learning.
understand biological development
Okay.
and other complex living systems, too.
Exactly.
And the sources are, well, they're broad. They cover physical models of single cells, fluidynamics.
Okay.
Then they pivot pretty sharply over to advanced machine learning techniques applied to really complex biological data like brain networks, even the genome itself. Wow, okay. So our journey together today is to navigate all this material, pull out those key nuggets of knowledge for you.
And help you see not just what's happening in this field, but maybe why it matters for understanding life at all its different scales.
Yeah, it's a deep dive into how, well, math, physics, computer science, how they're becoming really indispensable tools in biology now.
Connecting phenomena from, say, the mechanics of one single cell
Tiny level.
all the way up to the intricate organization of developing tissues and, complex brains.
All right, let's get started then. Where better to anchor this deep dive than with the project that kind of ties a lot of this material together, DevoWorm. It recently hit a pretty significant milestone.
That's right. April 2024 marked its 10th anniversary.
10 years.
Yeah, it actually grew out of discussions within the OpenWorm project, which started even earlier, back in 2011.
Ah, okay.
And the specific spark for DevoWorm, the initial idea, was to model development in C. elegans, the little worm, and focusing early on how to incorporate crucial cell lineage information.
And the sources even point to the exact moment, it seems. Issue 179 on the OpenWorm GitHub.
Oh, really?
Yeah, titled Add Cell Lineage Info to PyOpenWorm. It's kind of neat seeing those digital breadcrumbs for these big projects.
Absolutely. And that digital open foundation is really key because over the last decade, it's become a truly broad community effort.
Right.
Public announcements drew people in early on. They've participated in conferences like PRRG
Okay.
and the Google Summer of Code program, GSC. That's been a huge engine for growth, especially driving a lot of the machine learning work since about 2017.
GSC seems like a perfect fit for this, doesn't it? Structured projects, bringing new talent into open source.
Definitely.
And I saw mention of educational efforts, too, beyond GSC, like a 16-week Sign-Learn ML course.
Yes, fostering that pipeline, learning and contribution. It's vital for sustained progress in a field this complex and interdisciplinary.
Makes sense.
It's not just about the code. It's building the community around it. So websites like openworm.org, Slack channels, the GitHub repos, they're the central hubs for interaction, code, data sharing.
So the sources really emphasize this isn't just individual labs doing their own thing. It's about building a collaborative community, shared infrastructure. That insight alone is pretty powerful for understanding modern science, actually.
It really is.
Okay, now let's really dive into the science itself. Some of these sources tackle really fundamental questions about the physical nature of life, starting right down at the cellular level.
Indeed. And a fascinating approach discussed is using tensegrity models.
Tensegrity.
Yeah, like structures, think trusses and cables, that are stable because of this balanced tension and compression.
Okay.
They've used these, even a simple 3-cell construct, in something called finite element analysis, or FEA,
Wait.
to model cellular processes, like, say, the contraction of the actin-myosin ring during cell division.
Right, FEA. Isn't that usually for modeling stress on, like, static things, bridges, car parts? How did that apply to something so dynamic and, well, squishy as a cell?
That is exactly the core challenge the sources highlight.
Yeah.
FEA is typically designed for small displacements, linear behavior.
Right.
But tensegrities, and presumably cells, involve large non-linear changes. You pull on one part, and tensions redistribute through the whole structure in complex ways.
Ah.
And the sources specifically noted difficulties with certain software, like Console. Apparently, it treats cell-like elements as simple Slack cables.
Which misses the point.
fundamentally misrepresents the tensegrity idea, where cables are always under tension.
Ah, okay. So the analogy isn't just visual, it's that cells, like these tensegrities, get their structure and their response to forces from being held together by their internal bits and their connections to neighbors.
Precisely. They're supported by the network around them.
Hmm.
Shifting slightly, another area explored is fluid dynamics within soft biological bodies. They discuss viscous streaming,
Viscous stream.
the sort of ordered flow of fluid generated when these bodies move through or within a fluid.
Like a tiny organism swimming, causing little swirls around it.
Exactly that. And how the organism's body elasticity, how squishy it is, profoundly impacts those streaming flows.
Oh, interesting.
They compared simulated flows around, say, a soft torus shape versus a rigid sphere, showed distinctly different patterns.
Okay.
And this idea also extends to intracellular dynamics. Inside the cell, they differentiate streaming, which is more ordered, directional flow of cytoplasm.
Versus beating.
Right, which is more localized oscillation.
And there was that really surprising finding about simulated twisters inside oocytes, the egg cells.
Yes, the simulation showed these complex intracellular flow patterns slowly, over long periods, actually reorienting themselves towards the cell's main geometric axis.
No way.
Yeah, it suggests that the underlying physical dynamics of the cytoplasm aren't just random. They align with the fundamental structural symmetry of the cell itself.
That's wild. It's like the cell's internal weather system is subtly guided by its overall shape.
Kinda, yeah.
Okay, moving up in scale a bit, the sources also bring in physics metaphors for patterns and symmetry in tissues. They talk about biological hypercrystals.
It sounds exotic, I know.
Yeah.
But it's drawing an analogy from physics, specifically soft active materials, liquid crystal dynamics.
Okay.
They're not saying tissues are inorganic crystals, but using that concept of ordered patterns emerging from local interactions to describe how tissues organize. Methods like Voronoi tessellations get used here to quantify and describe the geometry of cell packing.
And how does this geometric organization connect to the big, large-scale body plan symmetries we see during development, like head, tail, left, right?
Right. So the sources discuss the emergence of those fundamental biological symmetries, anterior posterior, that's head tail, left right, and dorsal ventral, back belly, during development. They point to systems like the BMP pathway in organisms like Cnidaria, think Hydra, or sea anemones, Nematostella.
Classic examples.
Exactly. A morphogen gradient gets established, and then negative feedback mechanisms, they mentioned, one involving a protein called Z-SWIM46, in this case, help sharpen and stabilize these gradients. And that defines positional information.
So these axes basically establish a kind of dynamic biological coordinate system within the developing organism.
Pretty much, yeah.
Where the levels of these morphogens tell cells where they are and what they should become.
Especially in simpler systems like Cnidaria, that's a core principle of how large-scale patterns emerge from molecular signals.
Okay.
And within these pattern tissues, the sources also discuss models exploring cell mechanics, particularly how spatial constraints impact cell proliferation rate.
So literally just how much space is available limits how fast cells can divide.
Precisely. They link this to a mechanosensitive cell cycle checkpoint. The cell literally feels how much space it has.
Wow.
And this physical constraint isn't just a limitation. It can act as a signal contributing to cell differentiation. Well, when cells are packed tight, they can't proliferate rapidly, right? So that state might signal them to adopt a different cell fate instead.
Ah, I see.
And interestingly, cell division itself is presented as necessary for tissues to generate the forces and volume changes needed to, say, invade or move into new spaces during development.
There is also mention of how the cells connect and rearrange within the tissue.
Right, the dynamics of cell junctions. Things like T1 transitions in epithelial sheets, that's where cells swap neighbors.
Okay.
These rearrangements change the topology, the connectivity of the cell network within the tissue. And that affects overall tissue properties, like how much it can elongate or how fluid-like versus solid-like it behaves.
And then there's this concept that seems to challenge a more maybe traditional fixed view of development, lineage-independent differentiation.
Yes. This is a surprising insight if you're used to thinking of cell fate as strictly determined by ancestry, like a rigid family tree. It's well known in plants, actually, but the sources suggest it likely happens in mammals, too. It's the idea that a cell's identity and fate isn't solely locked in by who its parent cell was, but can be significantly influenced, maybe even changed, by positional signals from its immediate environment and its neighbors.
So context matters.
Hugely. They mentioned examples like wound healing or instances of de-differentiation where cells seem to sort of rewind their fate and take a new path based on the context they find themselves in.
So the cell's social network and its location can matter just as much, or maybe even more sometimes, than its strict lineage. That's a powerful shift in perspective.
It really highlights the remarkable plasticity that's inherent in development.
Okay, so we've looked at these physical models, how they describe cellular and tissue mechanics. Now let's transition to how computational power and machine learning are being unleashed on biological data.
Right. And this is where the scale and complexity of the data really come into play. Graph-based methods are a major theme here.
Representing biological systems as networks,
Yeah.
right? Interconnected parts.
Exactly. And the sources discuss hypergraphs as a way to go beyond simple pairwise connections.
Beyond pairs.
Yeah, to capture higher-order relationships, how groups of nodes interact together, not just pairs of nodes.
Where would you use hypergraphs in biology then?
Well, they reference their application in analyzing complex brain networks, for example, looking at how different regions of interest, ROIs, coordinate during tasks like decision-making.
Okay.
They did note challenges like ROIs potentially overlapping, and just the difficulty in visualizing and interpreting these really complex networks, sometimes calling them hairball models because they're so dense.
Hairball models, I like that.
But tools like Hypergale are apparently showing promise in navigating this complexity. They also mention looking for differences in network structure in conditions like autism using these methods.
Oh, that's a concrete application. And graph neural networks, GNNs, what about those?
GNNs are another powerful network tool. The sources touched on different approaches, like edge-based versus node-based GNNs, and their application in modeling systems like fluid mechanics. But more relevant here, they described how GNNs could be used to model a neural developmental program graph.
The network of neurons developing over time.
Exactly. The GNN could include processes like graph convolution, updating the state of nodes, the neurons, and even simulating graph growth as new neurons and connections are added.
And adjusting the connections.
Right, adjusting edge weights as connections strengthen or weaken.
That really sounds like it captures the dynamic, growing nature of a developing nervous system.
That's the goal, to learn and simulate those dynamic rules. Another technique mentioned is topological data analysis, TDA.
Topology.
Yeah.
That's the study of shape, right? How does that apply to biological networks?
Exactly. TDA analyzes the abstract shape or topology of graphs and networks. The idea is to find a way to summarize incredibly complex network structures using simpler, persistent parameters, like counting holes or connected components. The sources mention tracking how these topological shapes change over time in developmental data. They do frame this as maybe a bit of a reach goal right now.
Still early days.
Still early days, but it's a promising path to analyze network evolution in a way that goes beyond just counting nodes or edges, capturing the structure.
Beyond graphs, what other machine learning techniques are being brought to bear on this biological data?
Well, the concept of attention was discussed.
Not like brain attention, right?
No, the machine learning mechanism. It's used in models like transformers. It allows the model to focus on the most important parts of the input data while it's processing it.
And how is that useful in biology?
Particularly for analyzing vast genomic data. Attention mechanisms are key to models that can process really long sequences, helping to identify important features, patterns, maybe the grammar of the genome.
Grammar of the genome.
By highlighting regions that matter most in a given context, like gene regulation.
Okay.
Recurrent neural networks, RNNs, were also discussed for learning the dynamics of biological systems.
So learning the rules of how a biological system changes from one state to the next over time, like predicting the future state?
Yes, essentially. They specifically mentioned using RNNs to model realistic dynamics of the C. elegans nervous system and its connectome.
Right.
Tools like the neuron simulator, even synthetic data, are used to train these models. And the objective is always to minimize the error compared to how the actual biological system behaves.
Trying to match reality. And reservoir computing. That sounds a bit more abstract.
Reservoir computing, or RC, it's another approach for modeling complex dynamics like, say, the flow of information within the single cell or how a cell makes decisions.
How does it work?
Well, the model often involves representing cellular components as nodes with complex, often randomized internal connections. That's the reservoir.
Okay.
Input signals are fed into this reservoir, and the complex dynamics within it somehow produce the desired output. They did note, however, that RC can be quite opaque.
Hard to understand why it works.
Exactly. Describing it as kind of this magic makes it hard to interpret.
A bit of a black box for complex dynamics, then.
Precisely. Then there's multiple instance learning, MIL. That's another technique particularly relevant for biological data, where the labels can be ambiguous or uncertain.
Like trying to define a developmental boundary perfectly, or label every single cell type.
Exactly, those kinds of problems. Instead of labeling individual data points, MIL learns from bags of instances. You know something about the collection, the bag, but not necessarily about each individual item inside it.
Okay.
It learns the distribution or characteristics within that bag.
That seems incredibly practical for dealing with the inherent variability and, well, messiness of real biological data.
It's designed for exactly that kind of scenario.
Yeah.
And finally, the sources touch upon the rapidly emerging role of large language models, LLMs,
LLMs.
and more broadly, foundation models in biology.
We hear about LLMs for text generation all the time, but for biology.
Yes, they're being explored extensively, especially for integrating diverse types of genomic data. Imagine bringing together data on, say, chromatin accessibility, DNA methylation status, transcription levels, all sorts of stuff.
Okay.
Transformers and that attention mechanism we just discussed are key tools here. They help these models find structure and make predictions across vast stretches of the genome.
And foundation models is a bigger idea than just LLMs.
Yes, it's a term coined by Stanford HAI. It refers to these very large models trained on a huge breadth of data. It could be text, images, genomic sequences, whatever. And the key is they can then be adapted or fine-tuned for a wide variety of specific downstream tasks. So it goes beyond just language and points towards building these highly versatile base models for different scientific domains, including biology.
So these computational tools aren't just passive analysis methods anymore. They're providing powerful new frameworks, predictive capabilities for understanding biological systems. Let's shift now to some of the fundamental principles about biological organization and complexity that these models are helping us illuminate.
Yeah, a core idea that comes up is the very nature of biological complexity itself. The sources reference Stephen Jay Gould's concept of the wall of complexity.
A wall of complexity.
The idea that life started very simply, and over geological time it evolved towards ever greater complexity, sort of hitting this wall where simpler forms still exist, but complexity can generally only increase from the simplest starting point.
And the models help us understand how that increase in complexity is built, how we get from simple to complex.
Exactly. They explore different mechanisms that contribute to building complexity from simple rules and producing stable, long-lived patterns. Minimal models are incredibly useful here.
Like cellular automata, those simple grid rules.
Perfect example. Models like Rule 30 are just astonishing because simple local rules can generate incredibly complex, non-repeating patterns. They can even exhibit memory states. So this leads scientists to ask, well, could simple local rules governing cell behavior similarly give rise to complex tissue patterns? Reaction diffusion models, pioneered by Turing, Murray, Meinhardt, those are another classic class of models.
Yes, famous for generating patterns like spots and stripes on animal codes.
Right. They're based on this principle of local excitation and lateral inhibition. One substance or signal activates something nearby, but also inhibits it further away.
Okay.
These simple models can reproduce a vast range of biological patterns, from shell markings to digit formation in limbs. And the sources emphasize, while we often think of diffusing chemicals, the underlying physics can be different.
Oh.
Any system that has this kind of local activation, distant inhibition dynamic can potentially generate similar patterns. It's the dynamic structure that matters.
This naturally leads to the concept of the edge of chaos, doesn't it?
It does.
Yeah.
This describes a state where a system is balanced, sort of precariously, between being too rigid and ordered, unable to adapt.
And too chaotic and unpredictable.
Exactly. In biology, this looks like a balance between robust function, like your metabolism continuing to work despite small fluctuations, and being brittle, highly sensitive to perturbations. Dynamical models are crucial for analyzing how biological systems respond to disturbances.
And I guess in a complex network, if you perturb a really highly connected node, that could spread disorder very quickly.
Precisely. The sources note that network organization matters a lot, accounting for things like source nodes where signals originate and phase shifts in oscillating systems can make models appear more robust to perturbations.
Making them align better with biological reality.
Right, better than simpler models might predict.
This ties directly into another core idea, reciprocal causality and biological self-organization.
Yeah, reciprocal causality is distinct from a simple linear A causes B chain. It's more like a dynamic loop where elements mutually influence each other.
Everything affecting everything else.
Kinda, yeah. Biological self-organization is fundamentally rooted in this concept. Think about gene networks influencing the state of a cell. The cell state influencing its local environment and neighbors, which in turn feedback and influence the gene networks and so on. It's a continuous, integrated feedback loop, always receiving input from the environment too.
Dynamically interacting all the time.
Yes. And finally, the sources briefly touch upon viewing biological systems through the powerful lens of open systems thermodynamics.
Thermodynamics. That often brings to mind the second law, right? Systems tending towards increasing disorder or entropy.
And life seems to be the ultimate counterexample. It maintains incredible order.
Exactly.
But the insight here is that biological systems don't defy thermodynamics globally. They are open thermodynamic systems.
Open systems.
Meaning they actively maintain their internal order, their low entropy state, by constantly processing a flux of energy from their environment.
The energy flow.
This continuous energy input allows them to resist the tendency towards disorder seen in closed systems. It's a fundamental physical principle underpinning life's ability to build and maintain complex structures.
That's a really profound point. Life doesn't break the rules. It uses energy flow to create local order against that universal tendency towards disorder.
Precisely.
Okay, we've covered some deep principles and modeling concepts. Let's get specific now and look at some concrete applications, datasets, and tools mentioned in the sources.
Yeah, the availability of rich, diverse data is paramount, obviously. The sources discuss both microscopy data, visual information about cells and tissues, and numerical data. They reference specific datasets like DevoZoo.
DevoZoo. That contains developmental data from lots of different organisms, right?
Yes, it's a comparative resource.
Yeah.
Includes data from bovert, that's Ilianassa, a type of snail sea. Elegans, of course, fruit fly, zebrafish, mouse, spider, even Bassaria. It highlights the breadth of organisms being studied computationally.
A whole zoo of data.
Pretty much. Connectome data, particularly for the C. elegans nervous system, is also readily available through resources like Nomato.org and supplementary tables from key studies.
Integrating data across all those different developmental stages must be a huge undertaking.
It absolutely is. Aligning data collected at different time points, maybe even with different techniques, it requires really sophisticated computational methods.
Makes sense. A massive project mentioned is the Brain Atlas project. What's the scope of that one?
The goal there is to create a truly comprehensive cellular atlas of the human brain, looking at both adult and developing stages.
Wow.
And this isn't just anatomy. It integrates multiple levels of data, transcriptional states, epigenetic modifications, functional activity.
All layered together.
Exactly. And compares across species like human, non-human primate, and rodent. It's about doing a full cell census to understand the incredible diversity within cell types. And it revisits some of those challenges we talked about earlier, like dealing with overlapping brain regions of interest.
They also applied some of these image analysis ideas to a very different but critical medical problem: breast cancer imaging.
A fascinating application, yeah. They're using image reconstruction techniques on low-dose X-ray mammography data, trying to improve the detection of really small tumors, just 2-4 millimeters in size.
Catching them much earlier.
That's the hope. And this project faces some unique challenges.
Beyond just the low radiation dose needed.
Yes. While minimizing dose using focused beams from polycapillary sources is key, they also have to contend with the natural background noise of normal breast tissue, which apparently has a characteristic purlin noise texture.
Okay.
But maybe the most counterintuitive part is the image reconstruction itself. They're dealing with data gaps that require them to actively fill in lines from partial information.
Fill in lines, not remove them.
Exactly. It's the opposite of typical in-painting techniques, which are usually designed to remove lines or artifacts.
That is counterintuitive. You're actively trying to synthesize missing information based on the physics and expected patterns.
That's the idea. It requires developing entirely new algorithms tailored to this specific, sparse data geometry. Very clever stuff.
Huh. Organoid modeling was also mentioned as an application area, using lab-grown mini-organs for research.
Yes, things like modeling brain organoids. The sources note interest in using these for, well, maybe potential supercomputing applications or even voice identification research.
Really?
But they flag significant challenges related to material science, just ensuring the organoids are stable long-term and developing ways to effectively stimulate and record from them. Still a lot of hurdles there.
And bringing it back to the core projects, the sources describe the overall data pipeline and available resources for DevoWorm and OpenWorm.
Right, the workflow is kind of conceptualized in stages. Stage 1 is focused on image segmentation and initial data preparation, separating objects and images, getting the data ready. This stage is described as being relatively well developed.
Okay, stage 1 is solid.
Stage 2 is about deriving higher-level representations from that raw data, things like graph embeddings. This is described as less developed.
Still work to do there.
And stage 3 is the back-end infrastructure and visualization of these embeddings. This is where that topological data analysis work fits in. And this stage is currently the least developed area, indicating places ripe for contribution.
So they're explicitly laying out where the big challenges and opportunities still are for people wanting to get involved.
Absolutely. The GitHub repositories are the place to find these projects and contribute. And the sources mention other OpenWorm projects beyond just DevoWorm,
Oh, yeah.
like Cybernetic, which aims to simulate the worm's physics and its interaction with its environment, and WormSim, which focuses specifically on detailed muscle models.
So it's this whole interconnected ecosystem of projects, isn't it? Each tackling a different piece of the puzzle.
Exactly.
All working towards a deeper computational understanding of this model organism, C. elegans, and by extension, biological systems more broadly.
Precisely. It really shows how interconnected these areas are. You need the data, you need the physical models, you need the machine learning tools, and the underlying principles to make real progress.
This deep dive has covered remarkable territory, from the physical mechanics governing individual cells and tissues, all the way to the power of abstract computational principles.
And their concrete application to data sets from diverse organisms, even critical medical challenges.
It really underscores the power and, well, the sheer variety of computational tools and modeling approaches that scientists are deploying now to tackle these grand challenges of understanding biological development and complexity.
It's a huge toolkit.
And hopefully for you as a learner listening in, this deep dive into these sources has served as a valuable shortcut, helping you feel truly well-informed about this incredibly exciting, fast-moving, and very interdisciplinary field.
It really is a place where biology, physics, math, computer science are merging in some really creative and powerful ways.
So here's a thought to leave you with, something to consider as you process all of this. Given the diversity of these approaches we've discussed, from simple physical models like tensegrity to complex neural networks learning dynamics, how do the relatively simple rules or components captured in any of these models give rise to the breathtaking complexity, the robustness, the apparent purpose we see in living organisms? What fundamental principle connects, say, the mechanics of a single cell changing shape to the ordered fluid flow within an oocyte, or the intricate pattern development of a brain?
That mystery, right, how simplicity generates complexity, that continues to be the core driving force behind so much of this research.